There we go.
Yeah.
Oh, yes. Success.
Great, thank you. Okay. Hello, everyone, and welcome to this luncheon. I think most of you guys know me. My name is Majidi Rawani with ABMI Application Center. We're very pleased to have Hossam, well, let's say in an informal way, Dr. Hossam Al-Mu'ain and Dr. Helen Wagner from University of Toronto, our collaborators on Landscape Connectivity Project. They are here for the third time. I'm very pleased to give this very short introduction. This is part of the Ecosystem Service Assessment project funded by Alberta Innovate through Ecosystem Services and Biodiversity Network. And it's a collaboration with ABMI looking at landscape effects on Alberta's biodiversity. So we start with Hossam's presentation, and then hopefully we have time for discussion and also feedback from you guys in terms of application. Please, Hossam.
Lights.
Sure, yeah.
Okay. Thank you everyone for coming in, and I see some familiar faces that I met last time, and it's a pleasure to meet with the new people, and we can have discussions after we finish. And today I will give you a little bit of updates on what we have done so far since last summer on the project when we are working on the landscape connectivity effects on Alberta's biodiversity. So just little bit of introduction here, to put things in context. So we know the human footprints that, as a result of anthropogenic activities on the landscape, can really affect the structure and function of the landscapes. And these multiple examples of the human footprints, like urbanization, agriculture, recreation, roads, all these features definitely affect the landscape structure and function at certain level, which is definitely have some effect on the community composition on biodiversity. And why we care about this, or why this is important, is because these two things are the main driver of the ecosystem services on biodiversity. They are very valuable, they're very important, and driving the value for the ecosystem services on biodiversity. And thus, it's very important to study connectivity, assess connectivity, see how the value of the connectivity in the landscape can help us understanding more about the community compositions and biodiversity. So also the community structure in the landscape is a multi-scale influence of the environment. So by that, what we mean is when we look at the habitat patch level, the size is important. As you go, or as the size of the patch increases, you would expect a larger number of species and larger biodiversity. Among habitat, the degree of how these patches are connected is important. So if we look at this example in comparison to this example, this is an intact landscape, and this is isolated, completely isolated. So the isolation can be a factor here. As it's more isolated, you have less number of species than it's more intact, as in one patch. But if you consider also that the matrix between these patches are different, the habitat heterogeneity, these ranges of the species abundance and biodiversity can really change in between these two patterns. So this is the importance of the landscape connectivity or the fragmentation from this perspective. It's not that only, but if you consider also the habitat amount, so this is another factor that can contribute to the biodiversity and the species richness. And as you have more habitat, as you get more species and values for the biodiversity. So to put these all together in this context, if we consider these two landscapes, they have the same composition and configuration. But if you look at the habitat quality now, the low quality, you can think about it as the mediator of moving, or the individuals are moving from this site to this site, or from this patch to this patch. The quality of this habitat now becomes important. Then you can see that there is a difference between the two scenarios, even though they are the same. So from this, we can see that there is no exclusive effect of one of these different components here. And also, there is a big debate on which one is more important to each species, or the general idea of the biodiversity. So from these ideas, what we are doing, or what we are interested in, The ABMI data, the massive data that the ABMI have, new to North America, new to the world maybe, they've been using the different local scale variables to understand the species models, like for example the batch metrics and the land cover, land use, human footprints. But what we wanted to add to this to help us more understand about the structure of these communities is the connectivity. So what we do here, or our approach is, we are using a human-based intensity or the human-based resistance, the human footprint-based resistance to calculate these connectivity maps and then see how much contribution of these maps can add at the landscape scale of our understanding to the biodiversity and species composition. So our research objectives, the first objective was to get these maps done, right? Calculate these maps. So first we wanted to create these resistance maps and then calculate the connectivity maps based on that. And in addition to this, also we wanted to characterize some landscape context for the ABMI sampling. The second objective, after we calculated these maps, we wanted to test these maps with the biodiversity data. So you have the connectivity maps, but how to validate it, how to test it? We do this with the biodiversity data that we have. And today I will show you some examples of the responses that we got from the vascular plants and the birds. The next step of this, for this objective, is to optimize these maps. So as you will see in the results, we created 18 different maps, and these 18 different maps are different maps, and they are not the same, and we wanted to see which one of these maps is more suitable for certain functional group or for certain taxa from the biodiversity data. And one thing, or one normal extension of this work and this great collaboration is extending this to test how these connectivity can help us understanding more about the community composition and structure in urban gradient in Alberta. And this project is an extension that we got 2 more years of funding for doing this work through the Center for Urban Environment at the University of Toronto, with collaboration and some contribution for funding from the Geospatial Center from the ABMI. But this is, so we are not going to talk about this one today. We will cover a little bit more for the 2 first objectives. So this is just a project scheme and the timeline that we have. Objectives 1 and 2, creating the maps and testing the maps, are, there were some delays because of technical issues and computation capacities and different things. But finally, we got these maps done, and we started to look at some results on how these maps can help us understanding more about the biodiversity and the species composition for the vascular plants and birds. And then in year 3 and 4, what we want to do is to test this model on the urban gradient for Alberta, and then probably also optimize these maps and then see what other products, what other applications we can use and implement with this system. So going through what we did to create these maps. So the first step was to define first the intensity values, the resistance values, sorry. And for this, we used the species agnostic approach, as we know nothing about the species, giving values to different footprints based on the opinions from the expert-based people from the ABMI Geospatial Center. And then with that, we created 3 different metrics: the human footprints themselves, the intensity of human use for each of those, and then the average of these 2 metrics. In addition to this, there was an insight also that was given to the green area and the white area, more the forests or the natural areas and the more developed ones. And as you can see in the example here in the table, that different features were penalized differently or have different resistant values, either using the footprint themselves or the intensity of use between the green area or the white area. After this was done, so that was done for 101 different layers. These 101 different layers or different human footprints and the land cover data were compiled together and then were rasterized to end up with, to end up with one intensity or one resistance value for each of these 101 layers. By that, what we mean is that if you consider this is a landscape or this is a regular landscape and you have these different features, for each one of those it was rasterized at the 10-meter resolution, and then the assigning of the values of the resistance for each, and then combining them all in one raster that represents the resistance for each for the overall landscape, the wall-to-wall. After that, we added the value of resistance as well for the topography and for water. And for water also, we considered 3 different values for water resistance, either no water resistance or 50% water resistance or full water resistance. That decision came from also brainstorming and from discussions with ABMI people and with stakeholders from previous meetings. And that did add some new insight in these maps. So finally, when we add these topography and the water with different resistance, we end up with a wall-to-wall layer of resistance. And also this wall-to-wall was calculated at 10-meter resolution. And then this was rescaled or aggregated at the 100-meter and 800-meter for calculating the connectivity. This is an example of what the resistance map would look like. And what you see here is the polygon that delineates Alberta, and the buffer zone around it is a 20% buffer around the area. So when we put our nodes, we can exclude the areas that have high current just due to the placement of the nodes, and then end up with just the extent of Alberta for the current density. And what you see here is an example of this landscape. Low values are low resistance, high values are high resistance. And this is one example of one of the maps where we have the high or the maximum value for water resistance. As you can see, water bodies and rivers are very dark or black in color. This is a different like illustration for these resistance maps. We zoom into 2 different landscapes, one by the oil sand region and one the Edmonton region. You can see how these values or how these different values are represented in the map based on the same scale from low to high, low being low resistance and high being high resistance. So now we calculated this resistance map for the entire province. Now we want to calculate the connectivity maps now. Calculating the connectivity maps, we used the circuit theory approach, and the software that we use to do this is GFlow for computational reasons. This is the same as using different software like CircuitScape or Linkage Mapper in ArcGIS, but because of these computational issues, we have to go with GFlow for this amount of data that we are dealing with. And what you can see here is these are the nodes or the current nodes that we put around the buffer that we have. And then we start the software, and then basically what you do is you turn the background, or you turn the raster layer into a circuit board, and then you simulate the flow of the current in these different pixels, and then calculate the current density at the end of the procedure. This is an example of what a resistance map would look like and what a connectivity or current density map would look like. And as you can see here, probably this is one of the features that might be a highway. And then here you can see between the 2 sides of the highway, there is no flow. And then, for example, if this point is like a green bridge or something, then you will get more flow at this point as they are open or connected. Okay, so to go through this process and sum it up really quick. So we start with this resistance map that we calculated from these different human footprints. And then we run the GFlow or the circuit theory approach to calculate our current density map. And then after that, we standardize these current metrics so we can compare these different maps and highlight the biodiversity highways in contrast to the background. So what you see here in these colors, so the gray scale is low values of connectivity, and the colored from red to yellow is low to high connectivity values, and these routes represent the biodiversity highways or values of more connected routes. So how this can be used? One thing that we did is we calculated it at wall-to-wall to have these overall perspectives to the connectivity situation in the larger scale for the province. But then another way of looking at this is, let's say you are interested in a certain area of interest and you want to calculate the connectivity specifically between two different areas or two different protectorates. What you will do is you go back to the resistance map, and then you put your nodes at the sites that you are interested in, and then apply Gflow and simulate it, and then you get your current density map or your connectivity between these two points. So you can either work it at a smaller scale or at a larger scale, and now we have the data that can support this and the way to do it. So we started comparing these different maps. So for the connectivity maps, now we finished the comparisons at the 800-meter resolution. And what you see here is these different maps, the 9 different maps for the human footprint, the average of both, and then the intensity of human footprint, the intensity of use. And what you see here are on the x-axis are the values for the different water resistance. So no water resistance, 50% water resistance, and full water resistance. And these are volume plots that basically shows the distribution of these standardized values. And what we considered are values with 1 standard deviation or above, higher quality pixels or high values of connectivity, and minus 1 standard deviation on lower are lower quality pixels or lower areas of connectivity in the network. The upper portion represents the gradient of the high quality pixels or the contribution of these pixels to the network, and then the rest of the map with the the black to white gradient represents those lower gradients of the connectivity values or the current density values. These are the different maps. So as you can see, for each of those, for the human footprint, the intensity of human use, and the average of both, we ended up with these different maps at 100-meter and 800-meter resolution. So in total, we have 18 different maps. And as you can see, just by looking at the presentation of the color scheme, they look different. They contribute or they represent different ways of biodiversity highways in these different maps. So this comparison, this is another comparison between each pairs of the maps. So comparing all the different combinations of the map for the F and U and the average, and on this axis the average as well, the 9 different combinations. And what we do here on the upper diagonal is comparing map 1 to map 2, and on the lower diagonal of the matrix that you see is comparing map 2 to map 1. So first we took one map as a reference and compared the other to it, but also we considered comparing the other map as a reference and comparing the second one to it. So we have a full spectrum on the comparisons. And these comparisons are basically comparing the similarity or the percentage of shared high pixels or high quality pixels. So as you can see here, that we have there are some maps or there are some different combinations that are that have higher similarities and some that have lower similarities, and this pattern that you see can definitely show us that these maps have different stories to tell, right? So it's worth it to proceed and test these maps with the biodiversity data. One thing to stop at right now here, so currently we cannot really tell which map to use, which map is the best, because we are still undergoing this research, and we just got the maps done because of the computational issues and the power outages and all the technical problems that happened. But now we feel more confident that we have these maps done, and we started to extract the values for the connectivity and for the landscape characteristics at these secret spaces, secret sites, and then use the full data set for the biodiversity now to test the full spectrum, not just for the sub-area in the center of the province that we use to do the pilot study to see the patterns or the trends of the study or the responses of these different groups. So, and then after that, we will have to do the optimization of these maps to see which combination of these maps would work better to consider one of these taxonomy groups, or which one would be ideal for a global perspective if you want to use one map for everything. So this is still going, and that's why we don't have like the answer to which one is the best right now. So bear with us. Computational limitations, we discussed this before. One thing I just have to say without going through all the details, that this is really, really intensive computation, and we lost so much time of computation, actual computation time, because of some limitations due to power outage and unplanned power outage and different things. But finally, we got that done with the support of the SciNet and Niagara system. That's an amazing system if you ever wanted to consider a high throughput or high performance computing facility. That's an awesome place to use. Okay, now moving to the second objective. So we already calculated our resistance maps, our connectivity maps, and now what we want to do is to test these connectivity maps with the biodiversity data that is available with the ABMI. So we have the 1,600 sites scattered across Alberta, 20 kilometers apart, and probably you know this way, but much better than me. And in the middle here, we have this area that we just considered for the analysis, just to do the pilot study and look at the responses of the vascular plants and birds, which we will be focusing on for today's presentation. So our analysis included an RDA framework, a redundancy analysis, with our explanatory matrix being the multi-species responses for the vascular plants and for the birds. This is for the birds, and then we have the 3 different explanatory matrices. We have the 13 different variables that were used with the species models that covers the land cover and footprint, and then we have 2 other variables, batch metrics like the batch area and batch perimeter. And we added on top of that the connectivity values of these different spatial scales. The same thing was done with the bird species as well. And then what we got, or the results that we get, is the first one is the ordination plot. And what we want to see here, this is a site species ordination, and the axes that you see are just the directions of these different 3 explanatory variables. The directions of these variables can show that they contribute differently to the final variance that will be explained. So they have different contributions to the final variance. These are the results from the vascular plants. What we can see here is the land cover footprint explained about 22% of the variance in total. Uniquely to the land cover footprint was about 18%. The batch metrics in total was 1%, but a lot of it was shared with the other 2 explanatory components. And the most interesting part is that the connectivity, even though it has a 5% in total, uniquely it explained 2% for vascular plant at this small area that we considered. For the birds, even more. So the land cover footprint explained about 19%, 15% of it was unique to it, and then for the connectivity, it was in total 7%, but about 4% unique to the connectivity. And then finally, the patch metrics in total was about 2%. So what does that mean? So in conclusion, what we see is from objective 1 and 2, the biodiversity data that the ABMI have can be a very important data set to compare, or can be used to compare these different maps and to see the potential of each of these different connectivity models. Also, these connectivity maps can help us a lot, adding to our understanding about the biodiversity and the community structure of these different taxa. And what we are planning to do now is to test it at the full scale when we have the results extracted from these maps, and then contrast them to between each other and see which one is more important. And then all these products, all these resistance and connectivity maps can definitely help us in our future directions in promoting biodiversity and ecosystem services and planning. So for the future directions with the objective 3, what we want to do now is to optimize these maps and see which map combination at which scale will be better explanatory variable for which of these different taxa that we will be using. And then another thing is to compare this current detailed way of looking at the resistance and calculating the connectivity to a more simpler approach as used in Ontario region with Jeff Bowman and his team, where they considered only 3 different categories of classifying the landscapes to the degree of naturalness, whether it's natural and permeable, natural but impermeable, or impermeable to the movement of the species in the landscape. The implication of these, or the applications, the different applications that we can use with these maps actually is more important to everybody, and this is what we all think about, and we want to brainstorm about how we can use these maps for bigger potential. So one thing we were thinking about, there are 3 main axes to use these maps. One of them, and I will show some examples for this, is how we can use these maps to mitigate linear features in the landscapes, like highways or energy corridors. Another application is how to use also these maps to prioritize the conservation in the landscapes. So let's say we have different areas that we are interested in connecting or preserving, and in between these areas we have some places that we can potentially consider as important landscapes to maintain and to conserve, to preserve the landscape around these areas. Which one to prioritize, which one to give more value or to start with. And then the last thing that is very important, definitely and not least, is the biodiversity market, and how we can use this approach, how we can use these maps to rank these different landscapes, similar to what was done with the wetland ranking, as it was presented yesterday in the workshop. So I'll show some examples of this. So this is from Theobald and Dixon, and what you see here is just the looking at the intersection between the biodiversity highways and the linear features. Here, for example, was the energy corridors, and the importance of these application is that when you want to mitigate these linear features on the landscape, and you look at the intersection between these linear features and a major biodiversity highway or an important biodiversity highway, where to do the mitigation, how to preserve or conserve these highways not to get an influence on the biodiversity and the biodiversity market. Another thing is to look at the highways. So this is an example with the intersection between the biodiversity highways and the energy or main flow routes, and then here with the highways. Another example is to look at the prioritization of conservation in the landscape. So this is also from Dixon and Theobald and their teams. And what you see here is a resistance map. Is a resistance map with these polygons that represent the protected areas that are in this area of the Western United States. And then here, these are the points that were used to calculate this current density. And now only we focused on the areas that could be important to connect these. There's a close-up to it. So this is considering only the public land, and what you see in the green polygons are the protected areas. And these color maps show if you want to connect these protected areas, which ones or which areas of these public land have more potential or more priority to conserve, to maintain the global biodiversity or the global connectivity for these different areas. And then the biodiversity offset ranking is if you consider the change of the land use land cover for a certain landscape, how do you classify or rank these different landscapes, giving them rankings, for example, A, B, C, D with different importance? If you want to change or to replace a landscape that is in A class to a landscape with C class, what is the value that you want to consider? How to change these landscapes? What are the consequences of changing these landscapes based on the resistance values and the ranking of these landscapes? Of course, these different applications and could have some challenges. Some of these challenges relies in which is there a single, simple, best map to use? We don't know yet, right? We're still trying to test these maps. We have 18 different maps, and which one of these will be able to answer our questions for the global picture? We don't know yet, and we are trying to resolve this. This could potentially require recalculating some resistance values, right? Because if you consider assigning different values for the land cover type that you would predict that it will change to a different land cover, you have to go back to the resistance layers and manipulate the land cover map and change the resistance values, and then compute the final resistance value of the resistance map, then do the G-flow. So it's work intensive and non-trivial, definitely. Also, the comparisons, whether you work on a wall-to-wall scale or a certain area scale, what does that mean? If you simulate the current or the connectivity for the global map, wall-to-wall, or you do it for the smaller area, are they the same or not? Whether they deliver the same message or not? So what are these values mean? So these are all different challenges. Also, the local pixels, the values for the current, for the total current, whether it's the same considering a global perspective or it's different, and how this would look like if you change one of the resistance values. So all of these are challenges that we should consider while we are thinking of these applications. Assigning the values to the features in developed areas, that's another important thing, because if you have an area that is very populated or highly used, has high intensity of use, an area that is not that much used, whether you will treat both the same or they will have different values of resistance that can contribute at the end to the final connectivity picture or not, this is another challenge as well. So here is an example of what that would look like. For example, like if you have this landscape and you want to, this is a map of the current density. Let's say this is a highway, and if you want to mitigate this highway for some reason, and this need to be blocked, then you know that this is an important spot that can keep the flow between the two sides in this landscape, and then this is an important spot to keep in mind, not to mitigate or not to change or to preserve. So that's it for me, and with that I really would like to thank you all for having us today with you, and for listening, and for all the support, and for all the funding, and I'll take questions. Thank you.
And we hope there's not just questions, but a discussion. Where you want to see this go.
I have a technical question. How are you summarizing like narrow or small human footprint when you're up at like the 800-meter scale? If you have a road in that pixel, is that pixel a road, or is it like the density of roads in there?
So all of the features were calculated the original map at 10-meter resolution. Mm-hmm. And then to calculate the connectivity for the wall-to-wall, these 10 meters were scaled up to 100 meter. And then we used this to calculate or to do the G-flow stuff. So originally the data is there from the final resolution, and it's just rescaled to a little bit higher resolution, and then we use it to the G-flow. G-flow has one computation. This is a limitation, definitely, but G-flow has one like computational problem if it cannot handle more than certain number of pixels, and then beyond, like we tried to do the 10 meters connectivity and the 30 meters connectivity, but that at the full scale or at the wall-to-wall scale doesn't work. The GFlow is not designed to do.
How did you get from the, like, if you have, within 100 by 100, you have 100 cells of 10 meter. How did you take the average, or what did you do about those values?
Yeah, that was the average.
The average.
Yeah.
What, like, are seismic lines counted as like resistance in the maps? Like those can be like 2 meters, right? I think that's so. Like, if I don't know if you're taking the density of seismic lines in that one cell, or like if that cell just has a seismic line, it is cumulative.
I know that they were calculated at the 10 meters, so whatever values in these 10 meters for whatever features are there is the ones that I went with for the computation. If it's not in the 10 meters, then it's not.
I think those are specific, like linear feature that probably depends on the goal. You may need to go back and reconsider. But right now, it was just species free somehow. But if you have a species that basically react differently to a linear feature, like seismic line, definitely.
Then you can go back and modify all these linear features and all these components, and then it will be more specific, species specific than species agnostic.
Yeah, it might also be too detailed at that point if you're doing it for the whole problem.
To pick up a signal.
And still, I think, because we're thinking of now with the next 2 years, developing a few case studies to show how this method behaves. And I think the linear features really deserve special attention, whether that's roads or say seismic lines. How is that handled? Because,
Definitely, yeah.
So can I ask a question?
Of course.
Can you go back to the map comparison? I just want to know if you already see any pattern there.
Yeah, changing the, that was very interesting actually. Changing the water value, the water resistance value is really important.
you can see it here.
Yeah, I want to know if there is a specific pattern here that is interesting.
So here, once you change the, so can you do the light please, because I cannot see. There you go. So if you look at the water resistance here for the F, for the FU, and for the U, those three values with no water resistance are the least similar if you compare them to the others. So each one that has a 0, look at the pattern here. Each one that will have a 0 will have less similarity to the other ones. So manipulating the values of water resistance is very important. This is one thing that can change these values.
So does it suggest that we need more than 3? Like we have now 0, 0.5, and 1.
I think
we need to play more with this.
I think this can be answered when we test the biodiversity data with some insight into whether the water is actually a barrier or a barrier to these groups for movement in the landscape or not, because that can, you know, if you see like a contrast between a group that the water is not really a barrier and then a group that it is a barrier, then we can focus on one and then start changing the different values. And water values to change in the computations is not as difficult as changing values for the resistance for the 101 different layers, because this is one final layer that you can change its values and run the code, and that will compute a final resistance map, and then from it you can do the.
And this is based on 800 meters.
This is based on 800
not
meters,
100.
not 100.
So you may get a different pattern.
With the 100 meter, yeah, definitely.
And the partitioning biodiversity composition or partitioning is based on 800 as well.
On 800 as well.
Just this pilot.
Just this pilot.
This pilot is one map at 800 for that study area, the
Yeah.
smaller one, but we will soon have that for also the 100 meter for all maps.
For the different combinations.
Which combination of map did you take for variant partitioning?
Which combination?
Which map did you use?
That's the F map.
Yeah.
The F.
Okay. So there you.
There's still the U and the FU, and then the different combinations. And then what be interesting as well is when we start looking at the scales as well for each of these ones, with different groups.
So water clearly has an impact, I think, because of the amount of water we have in the landscape in Alberta. But I wonder from an ecological perspective if it's really acting as a barrier in a way that we're actually going to be able to see anyways. Like, because it mostly impacts, I would argue, rarer species, if anything. And only if it acts as a true barrier. And over time, I guess, is how I would think about it.
Yeah. It's kind of the question I had last time.
Yeah.
I think because they consider also the area, like the lake that will be has impact compared to the small pots. It depends on the species again, the species growth. It's just hard to say. Right now, I think I know they were trying to say this is a species creep because yeah, we can't basically just develop an Asian maps for many species. But then that's one saying that okay, maybe if we play with a little bit more water, maybe we get a different pattern. I don't know because right now we have 0.1 and 1. But I know that it's a lot of work basically. I don't know whether this makes sense to show this uncertainty basically at this level, or we just leave it like as a species free map. And then if we have a species of concern, for example, we can go back and we can basically really think that whether that species can pass the water or not.
This will never replace a species-specific analysis for a species of concern.
Of course.
But I mean, in terms of the uncertainty analysis, now we have the two extremes and the average, so like the 0.5 or 1 weight for resistance for water, and we have all the biodiversity data to see what does it look like. And the answer, I think that none of us knows the answer, and obviously it will depend.
Yeah.
I think at the end of the day, it's important that we are showing all the uncertainty.
Yeah.
This is something that probably you know that at least what is the range, and then you can play with this. I know most of the time when you present, they say, okay, just make an average, but it's good to have the real data and then decide which one you want to take.
So when you were doing the analysis with the plants and the bird species, I guess we could not talk about birds because most of them can get over water if they really want to, if they're motivated enough. But for the plant species, that group of plant species that was tested, how were they picked, I guess is my question. Because is it comprehensive? Is it everything?
Yeah,
it's everything.
Everything in there. All the vascular plants that were sampled at the plant points or the sampling points.
So there's no there's no
specific function group.
Yeah. Well, no. What I guess I'm getting at is that the plant data. There's some concern, I think, about some of that plant data in terms of its comprehensiveness. Like that, the sampling protocols and stuff have suggested that. It may not be truly representative of what is actually there. And so I wonder if it may lend some bias to the outcomes of this analysis.
I know there is a new data set now, like the most updated data set, I think for 2017, right, Ernest?
Yes.
Yeah, so this is...
But I don't think it will be nice. Of course, we know that we cannot solve the barriers, we cannot solve everything, but again, whatever we do in the landscape, we sample it. It's a sample. It's not to solve.
Yeah, but I think when we're looking at it from the perspective of whether there's a barrier to movement by assessing whether something is in one place and then is in another place, if it got missed in that other place, it is going to really bias the outcome of that research. And I think there's some question about that.
Yeah, then it will increase the uncertainty. But again, we are looking at the overall. It's not a single species.
Yeah, this would really be, of a concern, I think, if we are talking about a single species. And again, if you are considering a community, the change in the community, the community composition is different because it's one, you look at all the species at the same time in a multivariate context, not just one species.
Yeah, I'm just wondering if there's going to be bias in what's driving the outcome.
Definitely.
It will be variance. I don't know if
Well, that's
it's
a question.
bias.
Is it
Yeah,
just
it's a question.
variability or is it bias? So
Yeah.
are you more likely to detect
But
it
he-
in areas where we have high connectivity? No, you're just probably equally likely or
Yeah.
unlikely to detect the same plant no matter where it is, right? It's
Yeah.
just some plants are easier to detect than others.
Yeah.
And then the other question is, are those species that are hard to detect, are those the ones that, for example, respond differently to connectivity than the ones we do detect? Those are the things that could create bias.
I think when we discussed last time, if you remember, we said that, okay, if we get time in the future, maybe we can look at the plant traits. For example, dispersal ability of plants. Yes, it's different.
Yep.
So if we can add that traits basically to our analysis, then we can come with some of these basically concern about the design or find out more about this species or species groups.
Yeah. I mean, if you tell us this is species that are easy to detect, medium, and these are really hard to detect, we can do the analysis separately for that.
Yeah.
If you tell us these are the species that wind dispersed, these are bird dispersed, these are ant dispersed, we can test for that. What's hard for us is to find all the traits.
I think that's our push for that was a functional approach.
Yeah.
I think we're doing some work on it offline. We can talk about it. And there, one thing which made me curious about was in the RDA we have the explanatory power of connecting tools higher for birds than vascular plants, kind of counterintuitive. For me, at least, like, so I wouldn't say birds would be affected by connectivity.
More response.
I would expect less, but
it may.
I thought it's the other way because birds move, right?
yeah.
So when they move, and you base this on the resistance of movement in general, they move, like they tend to move more than the plants.
More, so they wouldn't be affected by the road or whatever, right? So anyway, you can
It may be though. It may be things that are that are associated with the measure of connectivity, but aren't connectivity itself that's driving that relationship. You know, like the avoidance of disturbance or things like that. That can be.
Or it might be a function of the scale of the analysis.
Or the
So if
scale
it's
of
on
the-
800 meters,
I
that
think we will get to
matter
that
for
100
birds.
meters.
800.
That's the 800.
Okay.
So I think all of these resolutions will like the clarity of the conclusions or the behaviors of these different things will come more clearer as we test the more extensive data with the full extent and with the different patterns of the different versions of the maps at the two different resolutions. I think we will have a very good idea or a better idea on what is more important to what.
What did you use as a response variable for the species?
These
level
are the
analysis?
abundance data that were collected at these sites.
Okay.
So trying to
the 0 to 4, not the predicted abundance or the observed abundance?
The raw data.
Okay.
The raw data.
But you didn't put in some of the predictor variables that we use in our models, for example, like climate variables.
No, I didn't. But this is one thing also that we want to consider. So there
Yeah.
are some other landscape variables that we like some other like batch-based metrics, like the effective mesh size and the edge density.
Yeah.
So
distance to edge, right?
distance to edge and distance to edge and effective mesh size.
Yeah. And
So-
climate too? Or
And the
no?
climates.
Yeah.
Yeah, these are. But what
Because
I used
there are some
only.
kind of key variables that we use that might, if you include them, it might better elucidate the variation.
Yeah, what I only included for this pilot study is the land cover and footprints and the patch metrics that we use for the species models.
So I think the general idea is basically to take the aerial speakers, basically call it ABMI, the models as null hypothesis, and then try to see if we can add
How much more you can
variation
with the connectivity.
using the connectivity maps and data that they have.
And more importantly, how this can contribute to, probably elaborate more on the models, or which one will be used to explain better version of the species.
My feeling is that it's basically you must be very brave to walk into this area because the data is vast, huge. In Alberta, basically, human footprint compared to US, they had 27, we had 100, 110, 115, basically. And basically, there's a lot of different discussion around different aspects of this connectivity and so on. But probably this is the most comprehensive connectivity.
I haven't seen anything in my life like this, honestly. Okay, so many data sets and data repositories, and I haven't seen anything like this. That's massive.
It is really interesting. I am curious about the possibility of doing comparison to like Jeff's simpler approach to that, and
I wrote that down too.
is that addition of complexity actually explanatory or not, right? Yeah.
Especially when you're thinking of landscape connectivity, like species agnostic,
Uh-huh.
and whether it's connected footprint or not. If you look at it from that perspective, I think we're looking at it from what part of our landscape that's remaining native is actually connected.
Mhm.
So, or flip of that is, where's the footprint and how connected is it?
Right.
That's sort of level. It would be interesting to see if we could get to that more simplistic level, and whether that's helpful, whether that explains what we're trying to look at or trying to report on for our specific project.
Yeah. For what we want.
For our specific project. For the world, just for our specific project.
I was wondering, so if we find there's similarities between breaking it down into those 101 features and just using 2 or 3 broad classes, would it be worthwhile to have even just 2 different resistance layers, like one saying these species move on a daily basis and ones that don't? I think the area we touched on, like birds have much different movement patterns than mammals, and even I get the resource to break down to every unique species is just not possible. But even just saying these are species that when they're moving, that's because of seed dispersal or some other mechanism, and these are the ones that are actually walking and flying and moving across the landscape, might elucidate very different patterns.
We can check that by grouping them and see how much of a difference it makes. But I think you can also at the same time, instead of saying people have species with different mobility, you can think about grassland species, wetland species, different types of species. That probably would explain more, right? But then you start making a connectivity model for different habitat types, and you end up with 1,000 maps in the end. So
this way, either you go one fits all, the species agnostic thing, and that's what matters more for the decision makers or conservation, or we want something that can work for everything. Or you want to go from the species size and then parameterize and optimize everything to explain the best. And where to meet in the middle, we have, I'm not sure.
You don't have it?
I don't know that I have it in mind.
I think we have.
I have something that,
somehow, because we don't know about all the species. Maybe we know about 25% of the species, but most of the species we have no data.
Yeah.
So we have to correlate with the species we kind of map, or at least the large group, like functional groups. And we can be really species. And there is a way that we can optimize what level basically species stay to keep enough information that can help us in terms of conservation, for example.
So what husband just went is something we discussed also in the conference in the summer. So what's the perspective? Do I look at the landscape from a conservation perspective? So I need to get the scale right for my species of concern, and it's different for every species. Or do I come from a land use planning perspective, where I have one multifunctional landscape that I have to manage, and conservation is just one aspect, but I operate at a human scale. It's like ownership of parcels and so on, political boundaries. That's what determines scale. And in between, we go like, okay, maybe there's grassland species plants or so that, they respond to the same landscape in similar ways. So kind of gradient. And then there's the other one. Am I looking at kind of the focus? Am I looking at just the natural landscape? Am I interested in humans as actors, which you're when you're with the biodiversity markets, right?
Mhm.
Or on the human impact of the human landscape alteration. That's when we just look at the effect of human footprint on the biodiversity. So in this project here in IBM, we are kind of in this upper half. And what's happening here? So over here would be the species at risk. Then ecosystem services is more on the left side. And in between, I guess, is biodiversity management. So which goal, where are we? And from that, the species agnostic approach essentially comes from here, from the land use planning side, ecosystem services fits pretty well with that. Whereas the multi-species approach is where you start like with a species-specific model, and then you select another species, kind of representative of another one, and you try to see how much, how can you find an agreement for a solution that serves multiple species. But how do we get from these few species to all species? So but that's the dilemma, right? You cannot, with one approach, do everything.
Yeah, but it also does, I think that we run into the problem from a management perspective that that we want to do both,
yep.
And that they conflict sometimes. Yep. Like they don't always give you the same answer to that question, right? So then what do you do? But sometimes there's this is a clear choice.
If you do biodiversity of the number of species, that's two species are not the same, right? Like some are species of concern for whatever reasons, and some are just generalist ubiquitous, and you don't need to worry about them. But if you do biodiversity assessment, then it's the same currency. Mm-hmm. So it's a bit the same thing. This approach is not going to work for your specific species of concern necessarily, but it gives you. Mm-hmm. I think when we had this big discussion 2 years ago, connectivity is such a species-specific thing. How can we do such an approach? But then, by and large, probably most species are more happy in a natural landscape than in a highly built-up landscape, most of the species. So there is a shared response.
Pretty much most of the species.
Highways usually are not so good for species, right?
Yeah.
So there is a shared response that we're trying to capture. But it cannot capture everything, right?
So I just want to know, because you're also looking for feedback in terms of application.
Yep.
We want to get it from this stage to application. So what is your suggestion in terms of moving from this stage to the stage that we can really apply? Do they need to go with what they have planned, like taking ABMI data, optimizing, looking at the quality of different labs and answer that question? Or you have a different suggestion, basically taking a different approach that might gives you a better idea about what you need for application. Because right now we have this, they have a plan basically to go and test with the ABMI biodiversity data, and you're talking about the group, functional group, about taxon and different things.
That's essentially testing these models.
Models, basically. Yeah. And now we're looking at, okay, we have 18 maps or we have 36 maps because we have few scales. Mm-hmm. How to get to the point that we say, okay, we want to take this and that, this map, for example, that might be interesting for example.
Yeah. Yeah. Well, so there's two things that I'm thinking about from that perspective. So one of them is that a single map would be our best possible outcome from a use perspective within the biodiversity management framework. But I also think that when we implement biodiversity management frameworks, the actual action has to happen at a much smaller level than a regional scale. And so that the question you were asking, I think about it, when we look at things from like that higher landscape perspective, is it still applicable at a local level? Mm-hmm. Those are really important questions to us, because that's going to be the thing that we actually have to act on. And ensuring that there is a relationship between those things is really important, because otherwise we may act at a local level but not see any impact on the measurement that we're taking at that kind of regional scale, and that would be problematic. So
Contrasting global scale to regional scale.
Yeah.
and what that means.
yeah, understanding that relationship, I think, would be really beneficial.
So you need to be able to document that the action taken at the local scale to kind of maintain or improve that connectivity will actually improve it at the provincial scale. Is that what you're saying?
Well, not so we function at regional scales, not at the provincial scale, but yes, that's essentially the relationship.
And we're reporting at regional scales.
Yeah.
So we can get busy and do all kinds of things at a local scale and feel that we're doing all kinds of great work, but it may not translate, depending on that relationship, to that regional scale reporting that we're doing. So we can be really busy doing all kinds of things, but we're not improving anything in our reporting
Yeah.
because
Which is
there's a
problematic.
disparity, which is very problematic.
Yeah, from a political perspective.
Yeah.
And but yeah, on a
And what would
staff
be,
perspective,
what kind of actions are you thinking about right now where connectivity play factors in?
land disturbance planning, so cumulative effects management,
Yeah, really, like
protected
living
areas.
footprint management, yeah, those footprint management,
Yeah.
so linear footprint or footprint management plans in general.
So for an example is we might have a mechanism where we can identify native grasslands and use avoidance as a management tool for industrial footprint to try to maintain that native grassland cover. Avoidance is our first choice, minimal disturbance is our second choice, to try to maintain that native cover.
For conversion to industrial also.
Right, so to control the density of that industrial footprint with the objective of managing and maintaining the native landscape.
Yeah. And then if we could show that this would help maintain the connectivity
Mhm.
across the region.
Mhm.
And your regions is
The land use framework.
Yeah.
planning.
Just 7 regions.
7 regions.
Seven regions.
It's huge.
Yes.
And they span very disparate habitat types.
Eager
Natural
types,
subregions.
yeah.
From mountain to plain area.
Yes. Yeah.
Yeah, we have some that are way more diverse than others.
Yeah, that's true.
Yeah. But that gives us some guidance for the case studies. Kind of,
Yeah.
to see, okay, what kind of scenarios, and then at what's the largest level at which we need to evaluate that, and that is the contribution to your regions.
Yeah, another example, I guess, is protected areas.
Mhm.
And so they are almost, like an analogy would be postage stamps within the landscape.
Mhm.
So managing to sustain those protected areas, managing the matrix
Yeah.
surrounding those protected areas and the disturbance in those is very important to sustaining those small protected areas. that we
And that
have.
would be a classical kind
Yeah.
of application for this. Yeah.
Yeah.
And which region are you thinking about for that most?
Which one?
Well,
Like NSR is the one that we have the
NSR is-
most challenge with at the
Yeah.
moment. but-
NSR is?
North
North
Saskatchewan.
Saskatchewan, sorry.
So a little bit, so sort of south of where your study area was
Mm,
more, because within the North Saskatchewan, that's probably the region with the most disparate or most diverse ecology from west to east border,
Yeah.
across quite a few natural subregions.
Yeah.
A lot of white area land
Mhm.
compared to the other planning regions.
And also, I
And
think
a lot of green area too.
the biggest diversity in footprint as
Yes,
well
yes.
in that region.
And yeah, the least amount of native left too. Yeah.
I think you're on the right track too, and that you're
Mhm.
considering in terms of applying or testing the application of the connectivity maps to the modeling of the ABMI species data. So if it looked like including these kind of data would enhance the models, and that's likely something that we want to do. So, but it's not as maybe a direct application is what we're talking about here. But there is a need for a connectivity map right now. But it could feed into other products too. So I just wanted to say don't lose sight of that either.
No, I mean that one is a given. We've been
Okay,
doing
yeah.
that anyways. And it's partly what we had hoped to be done by today, but it's going to take us a little longer. But with the additional funding we have now, with Center for Urban Environment and some supplementary from Geospatial Center, we promised to develop some case studies. Now we want to make sure these case studies help you guys. Whereas it has to be on a urban gradient for the main funder, but we can choose these within
North Saskatchewan.
North Saskatchewan.
Let's do the North Saskatchewan
Yeah.
plan again.
Yeah.
That would be.
It's got Edmonton in it as well
Yeah,
as
so we can
rural.
have 2 or 3 sites in there, and
got the
and then
significant
also look at the
forest
horse. Yeah.
management.
So
that would be-
probably next visit, then we can discuss more detailed kind of scenarios for this. So that's helpful for us.
Thank you. Any other comments?
Is there anyone on the phone with any comments?
They're not on the phone. It's a webinar.
It's a webinar.
Sorry,
guys,
we
we can't take questions from you. I don't know.
See if there are any.
Yeah.
And now I don't even know if anyone would have stayed on, if they could hear.
Yeah.
So they can hear us.
They could hear us,
I
yeah,
think they
but
can, yeah.
they
So in your comparison chart, I saw
look like they have any questions.
the analysis of the effect of water in the analysis. What about topography? That was another variable.
No
that-
question.
It's fixed
Oh.
to the
What's
every.
this?
So it's one topography resistance values that was applied to all the maps. It's the same.
But the impact is very low compared
Yeah.
to.
Yeah.
To
And you
the-
took the one from Theobald, right?
From where, sir?
Who is this?
The paper from
The-
Yeah.
from
the
US.
Stephanie
paper
Ball.
from, T-Ball,
What they're using in
what they
US.
used for.
So the effect is not so dramatic. I don't think it would make a huge.
They could hear us?
I think
Yeah.
it's.
So they're,
1,000
I don't know.
something. It's a very small impact.
And
I don't
it's-
know.
Mice?
Except
Mice?
for the Rockies,
Yeah.
it's gonna
Ah,
make a
Oh,
big
good
difference.
eyes. I can just see there's a question.
there are,
Oh, that's- I'm using my graduated lenses.
Like
See if I
that?
can get
No,
into
you're right,
focus.
Mike.
Can somebody read it aloud?
it says, have you considered the mite data? They are dispersal limited and connectivity could be particularly important for gene flow.
We will be
We
doing.
would
We
like to
collect-
look at them, yes.
Yeah.
We will be doing that.
I think lichens, probably.
Yeah,
mites.
bryophhytes.
Yeah, definitely they are in the list.
And
They're kind of, everybody thinks it's a caribou, right? But we're doing like things where nobody
Yeah.
thinks it matters.
So any other?
there are two more.
Yeah?
There was one that says, have you considered creating a model for wetland species to test against our wetland data?
Well, wetland connectivity is a whole different thing
Yeah.
because it is more linear. And so we've been talking with Monire Pharmacy yesterday, and I mean, there is interest, but it's like, that's a whole new chapter. So right now, we stick to terrestrial stuff.
Yeah, especially probably would be hard with surface water groundwater interaction.
Yeah.
Which is mostly about the temporal wetland and so on. So it's just a completely different area, but we were thinking maybe using the product that Geospatial Center produced, basically, the multi-temporal data, that maybe we can use that to address some of the connectivities. That might be very interesting. I'm very much interested, but I don't know how many days more I stay with ABMI, honestly.
Days.
if I stay, if I have a chance to work with ABMI, opportunity to collaborate with ABMI, I try to see if there are basically opportunities to go to that direction as well. I really like the idea because we have great data from the wetland. Basically, we can look at this, like looking at the broad species and just some connectivity.
There was one last question.
Yeah.
It says, have you considered the implications of your research to the spread of wildlife diseases like chronic wasting disease or the spread of invasive species?
That would be very interesting to look at.
The quick answer, no, we haven't.
No, we haven't.
And there's always a dilemma if you increase connectivity, invasive species and disease and so on, of course, will profit as well.
Of course.
So yes and no. Like I think that there's some other ecological things that happen when you have higher connectivity in terms of support of native species. That native species populations are sometimes able to maintain themselves better, and then resist that invasive species. And I think footprint, like from my perspective, footprint has a lot more to do with the influence of presence and distribution of invasive species within the province. Chronic wasting disease, I think, is an interesting case. I'd have to think about that one some more, whether I thought that was, yeah.
One example that I can think of is the Lyme disease with the deers, the movement of the deers, and the mites, the deer ticks. That's one example that I can think of using this, and that's why at the end of the presentation I was thinking of that these products that we have available now can have so much different applications and extensions to use the current data that we have and these products to test different things, like for example the epidemiology of different diseases in the landscape and at different scales with using these different maps that we have. So that was one of the potential areas as well to test these maps in. But
yeah,
it definitely goes back to resistance value.
Again,
Oh, who asked it? Okay, so did you already ask it?
No.
I think it's
invasive.
Stephanie Ball.
She's in the monitoring.
I see. We had some discussion last year with Victoria Wagner, who is working on invasive species here. She's a new assistant prof at biological science. And I had a chat recently with her. She's still interested to see, but she's considering all the roads basically good for invasive plant species, which is true, basically, because they can establish along the road.
Yep.
And they're basically,
and they're typically good dispersers.
Yeah.
So yeah.
That's
So it
one
depends.
important trait that makes them invasive.
Yeah, and diseases like the chronic wasting, for instance, is interesting because it's present in native species, right? And so, and the more I think about it, the more I think that connectivity would actually be a really interesting measure for that, because we've seen the movement of chronic wasting and mule deer along river valley corridors.
Because of the density of animals. Right?
Yeah, because of the density of animals, right? And so I think that, I think it could be really closely related.
Because of the lack of habitat.
Well, and also because that's where
forced
they,
together.
like, muleys will come into, like,
Well,
that
yeah,
area
they-
in the winter, and like, so
Yeah.
they
So
end up in
it
closer
might be that
proximity
this our biodiversity highways kind of thing can explain or predict where it spreads most quickly, but it might be.
It would be interesting from a management perspective in terms of, like, if you were going to try to do some of the control work that's been done in the past or is being done in some of the states right now, is there places that should be targeted because of that potential relationship? Yeah. Super interesting. Nothing related to anything I do right now, but really interesting.
But these are all interesting questions in terms of science and in terms of application anyway. And I think getting access to even to more databases, like I know that this bird database that Peter is working from the Boreal BAM, is it BAM?
BAM,
Or going to multi-source data and look at data from riparian ecosystems, or even using the ARU camera data maybe at some point for mammals would be, or for birds would be very interesting basically.
Some human footprint types increase connectivity for some species and decrease for others. I think that's a problem.
Yes.
Yeah, it comes again to the species.
So really
question.
worry about managing the stuff that increases.
Yeah. I mean, wolves like to move
Until it
in
becomes
Algonquin,
a-
they move along the
Sorry,
roads.
not species that take tax like plants.
Oh,
I
yeah.
was just thinking that, plants might actually be more connected by roads.
Yeah, and those are the plants that could be.
dispersal of
It depends on
invasive
how they
plants, for
disperse.
instance, is
Yes.
much higher.
So I think this is a very
because
good.
of human movement, but also because of
I think this is a very
Is that
good
just more
discussion.
error in the
Yeah.
data set, or is that?
No, that isn't murder. That is, like, it can be tied mechanistically to what happens along it.
Yes.
But if this kind of approach is assuming there's a negative effect. Like, it's
So it
assuming that
depends on
all-
which taxonomic groups you're looking at, right? So.
But you'd almost, like, in an ideal world, you'd have different resistance values
course, yeah.
for different footprint types. maps depending on the tax.
Yes.
Well, but that's going again to looking at
Yeah,
a
yeah.
species by species or taxonomy.
But maybe it would be very helpful basically to have your opinion about the list of the species that they're working with. Like you can say these are sensitive species compared to this species. These are well dispersed species.
Yeah, if you give us groupings, we can
If we
test
can
them.
continue this discussion and
Well,
come
there
to
are
some
very-
groups,
Yep.
maybe they can test all of this
Yep.
and then we can see whether that makes sense or not. Because right now we just consider all the species the same.
Which
And we
ones are
can go
more
with very general categories
interest?
based on your
And
experience.
then it would be interesting to see
And I
is,
think it depends on how you want to.
Yeah, because definitely taxa is too broad. I think that even within a taxa, like several plants have no mechanism basically.
Well, yeah, there's generalists and there's specialists, just like there are in the animal
Yeah.
world.
yeah, that will make a big
And
difference.
that's- But then there is also the concept of ecological range, right? So expecting something to be ubiquitous across the North Saskatchewan is not realistic. It's ecologically, it's not possible.
Yeah.
It's not appropriate. So to have that weighted the same as, or to not consider their natural ecological range
Yeah.
as part of that modeling would be a mistake, I think.
Yes.
Which is maybe where natural subregion concept would be helpful.
Yeah. But that's like because the models, the ABMI models that we take as a null, they essentially have that in there, right? If it's this type of habitat, then this plant is not likely to be there. It's not so much the range versus the site conditions, I guess.
Yeah, because there is no real trait data in the model. Yeah. But I think this would have a lot of values basically. Just maybe we can think about this in future, have a group of people like your experience, take your experience, come with some general categories and then
And it would
test
be
really what is
really
going
nice
on.
then to see is like dispersal mechanism versus some other classification, which one explains most, like has the difference between the species responses. That would be actually really interesting, I think.
I can be so from generalists to specialists, for example.
Are you planning to publish some of this work, and what's your timeline?
So
The first one is almost done, like everything we need to have it is ready. It's a matter of putting it together and then submitting.
So that is just the maps and the resistance
the maps and the
values.
resistance values. And then when we finish the extractions for the secret sites and test the all biodiversity data, we will be ready for the second one, which we are already developing the code, almost developed a lot of the code for running the full extent analysis, and then we should be ready for the second one.
So sometime next year.
Sometime next year.
Yeah.
So there was one final comment, if I may share,
Oh, sure.
from Andrea McGregor, who said, "I don't think we are talking about increasing connectivity, are we? It seems like we are looking at how to plan for the maintenance of connectivity in developing landscape."
Oh, definitely.
Yeah.
It's the legal part of this connectivity issue.
But you were mentioning that in the North Saskatchewan, you got these isolated protected areas. And there's an interest in thinking how we can increase the connectivity.
Well,
you
you
have to
need to
maintain.
increase in
Maintain.
some areas within a region if you're decreasing in others.
Yes.
So,
Yeah.
but the biodiversity management frameworks have as an objective to maintain.
Yeah,
Yeah.
maintaining
At
and
a
not
regional
not
scale.
increasing or
Yeah.
decreasing
Yeah.
connectivity,
Yeah.
but increasing connectivity.
Oh,
Well, but there
can-
may be certain like local scale places where
Yeah.
that is a thing that does.
If you think of compensation, right?
Yeah.
We took something away here, then it would make much if we can compensate something somewhere where it really makes a lot of sense. Lots of
And I think
sense.
the caribou work speaks to that, like that they will be increasing connectivity in one way or another with some of the work that they're doing around reclamation and some of the linear features and stuff.
Yeah, that puts
yeah.
a whole other assessment of success of reclamation.
But if you want to do it in the south, it's a lot of
Yeah,
legal and a
there's-
lot of policy and a lot of
Well, and a lot less
Critiques.
success at
Exactly,
reclamation or
less success.
restoration and
Yeah.
grasslands.
So definitely we can suggest,
especially
but then
moist
the
grass.
application
Is
and
something that
increasing, that would be another piece that
Mhm.
I don't know much about that, sorry. I'm just, I have a feeling that based on my previous experience, that it's not that easy, basically.
Yeah.
To ask people. We were in a meeting this morning about green wetland, basically, that how difficult it is basically to convince people to.
Well, especially when you think of it